Can you hear me this time? Okay. I'm realizing that when I set up the Zoom meeting, I think I did something to the point of where I can't start the meeting until right before class starts. And it asks, and when I try to get it, it says that it's waiting for the host to start the meeting. Well, I am the host. So I don't know what's going on, except for I probably did something when I set it up that messed it up. So that I can't start it until right before class.
A general question. Do you teach AMP2 by any chance?
I don't know yet. I will probably, if I teach it, I'll probably teach either 2 lectures and 1 lab, or 1 lecture and 2 labs.
Okay.
And I haven't, because I'm an adjunct, I haven't been told what classes I'm teaching yet. I haven't even been shown the options.
Okay. Do you usually know when that takes place, or is that just kind of randomized?
They let me choose once the full-time faculty have decided what classes they're going to teach.
Okay.
And you're asking me, and the person who determines the science, purchases the science textbooks, is asking me also. And
Gotcha.
so I will try. I don't let me.
The only reason I'm asking is because since we're in like class now, we get first dibs over people who are just starting hack. So we get it's like at the end of October that we can already register for next semester ahead of everybody else.
Let me, if I have a chance, my schedule is really a little insanely crazy right now.
That's okay.
Well, let me just say, if I get a chance, I'll email my chair and figure out when he's going to, if he knows when he's gonna have things ready. Because if I think my schedule is crazy, his, I'm sure, is about 10 times as crazy.
Okay.
I don't know if when he sleeps, if his schedule is that crazy. So when I find out, I'll let you know.
Okay.
And if I can find out before the end of the month, I'll let you know.
Okay, sounds good.
How are you doing? How are you, Caitlin?
Oh, I'm good. Tired, but I'm here. Works a lot, but
I understand that.
Just going through the motions, I guess.
I understand.
I emailed you too.
When?
The first week in I emailed you a while ago.
Did I not respond?
No, it's okay that you didn't though. Because I emailed you and I CC'd you and Professor Bacon on the same email.
Okay.
The first week of November, I'll be in Las Vegas.
Okay.
But I'll be attending class. I think I figured out the time issue, so I'm not gonna change my time on the computer.
I think I did get this email. I'm almost sure I got it.
Okay, and it should let me in. So I mean, plane times are a different story because we leave Tuesday. We're like leaving Tuesday morning at 6, but we'll be in Vegas by 10. So I mean, either way, I'm gonna try and make class. I know they're a 3-hour difference, but
okay. Are there any questions or concerns?
I don't have a question or concern, but I'm not feeling the best, so I'm just gonna have my camera off today. Is that okay?
That's okay. It's
Okay,
okay.
thank you.
I understand. You look
Thank
like you're a little tired.
Yeah, I'm sick.
Okay. Other questions, concerns? My midterm grades are due within the next week. And like I was just telling Caitlin, my schedule is a little crazy right now. And that's really the major reason why, is because at all three of the schools that I'm teaching at, midterm grades are coming due. With that in mind, by the end of today, we should either finish chapter 3 today or be within a couple slides of being done. So you should have for lecture, you should have the SmartBook assignments for chapters 1 through 3 done by the end of this week. You should have the chapter practices done and the chapter quizzes. And be ready to do the first exam. If you're missing any of those assignments, that's going to bring your grade down. Midterms do not go on your final record for the school. They are only kind of a heads up notice to you as to where your grade sits at this very moment. D2L, the grade that D2L calculates for you can be a little misleading because it only gives you points for the assignments that you've turned in. It only tells you your grade based on the assignments that you've turned in. So for example, unless I go in and I put zeros for your grades, which I only do after the due date for something, it may give you a grade of a 96, but if you've only done 2 assignments, it's only giving you a 96% for those 2 assignments. It's not taking into account any assignments that you haven't turned in until I put zeros in there. And I have to manually do that. So I can't say I do it after the due date for that assignment, but I can't say immediately afterwards because it's not immediately afterwards. So make sure that that's where you should be right now. Is you should have all of chapter 1 SmartBook, chapter 1 quiz, and the chapter 1 practice done. You should have them done for chapter 2. You should be done with chapter 3 or almost done with chapter 3. Also, the first exam is posted. You have until the 20th at midnight to take it. I just got on and looked and realized that 3 people have already taken it. Good job. And I will tell you, I'm really happy with their scores. All 3 people scored within the 90s. You know who you are, so I'm not going to say any names. That's pretty good. It's really good, actually. So please get that done. Where is it located? Let me see here. It's located right here. So it's through McGraw-Hill. It's located right here. Any questions? So jumping into finishing chapter 3. So organelles of the cytoskeleton, or one of the organelles, the cytoskeleton, what is it made out of? We have microtubules, microfilaments, and intermediate filaments. Microtubules have rods with the largest diameters. They're hollow tubes made of spherical tubulin subunits. That radiate out from the centrosomes, which contain the centrioles. These microtubules determine the overall shape of the cell, the distribution of organelles, and chromosome movement during cell division. These are essentially the roads that motor proteins move organelles upon. This is the road system within the cell. And it is a magic road in that it's continually assembled and disassembled. It would be like deciding you're going to go to, deciding we're going to go to, where's it? New York City, and designing a road immediately to get us there the fastest. And then once we work to New York, we may break down that road that we just got there on and recreate another road. So this is our map system. Microfilaments are the rods of the cytoskeleton with the smallest diameter. Rather than being made of tubulin, they're made out of actin. Actin is involved in cell motility and changes in the cell shape and strengthens the cell surface. So this is the major part of the cytoskeleton involved in maintaining the cell shape and allowing for cell motility and keeping the cell surface strong. They also, because it's involved in changes in the plasma membrane structure, these microfilaments interact with myosin to form the cleavage furrow during cytokinesis for cell division, and attach to the cell adhesion molecules to produce the crawling movements that are involved in amoeboid motion, or as allowing cells to move along a surface or move past other cells by the formation of what are called pseudopodia. Pseudo meaning false, looks like, but it's false, and podia meaning feet. So these are false feet. Pseudopodia are created for movement. They're also created to allow for endocytosis and exocytosis. Like the microfilaments, or excuse me, like the microtubules, the microfilaments are constantly being broken down and reformed. and can interact with myosin to produce the contraction of muscle cells. So microtubules, microfilaments, and intermediate filaments are also part of the cytoskeleton. These are woven ropes that have a high strength to them. They attach to desmosomes, which are cell junctions, and act as guy wires to resist pulling forces. In nerve cells, they're called nerve filaments, and in epithelial cells, they're called keratin filaments. So just a small comparison here. Microfilaments made of actin subunits have the smallest diameter. Intermediate filaments are tough, insoluble fibers. Microtubules are hollow tubes made of tubulin. Microtubules are anchored by centrosomes which are granular matrices within the cytoplasm. Within each of the centrosomes, or within a centrosome, is a pair of barrel-shaped centrioles that are right angles to one another. Each centriole is a set of 9 triplets of microtubules with a hollow inner tube. So 9 triplets, 1, 2, 3, 4, 5, 6, 7, 8, 9. And each one is made of a triplet of 3 microtubules. So remember, these are what are creating the microtubules. So centrosomes contain centrioles, and the centrioles make the microtubules. Centrioles are a little underrated until they don't function. When I say that, because a lot of cancer treatments are targeted at centrioles, because they block microtubule function in centrioles. And if you know of anybody or have ever seen anyone go through cancer treatments make people really, really sick sometimes, a lot of times, because they're not specific just for cancer cells. So any actively dividing cell is oftentimes targeted by cancer drugs. One of the things that's very important for cell division is microtubules, because it forms the mitotic spindle, which allows for the separation of the chromosomes into daughter cells. Centrioles also form the basal bodies of cilia and flagella. So if you look here, here's our triplet of microtubules. Towards the top, the arrangement changes from going from 9 sets of triplets to going to 8 sets of doublets with a pair in the middle. This is specifically looking at a flagella. So the basal body is this right here. Towards the top, or towards the end closest to the cell structure, it looks like this, a microtubule doublet. So cilia are whip-like motile cellular extensions that occur in large numbers on cellular surfaces. They have an immune function. They move things in one direction across the cell surfaces. Cilia line the respiratory tract. They also line the fallopian tubes, allowing things to move easily across the surface. When central projections are significantly longer, they're called flagella. Sperm are the only human cells with flagella. Cilia propel other substances across the cell top surface, apical surface, whereas flagellum propel the cells themselves. There's a pattern of microtubules with 9 doublets encircling one central pair. We saw right here. Or 8 doublets encircling a central pair. How do cilia bend and move? They're very flexible. You've seen flagella, they're really great little swimmers. What allows them to swim the way they do? The individual doublets have arms or have motor proteins that act as arms that grip the doublets and start moving along their length. One motor protein on one doublet is connected to the doublet on another adjacent doublet. As the motor proteins move along the adjacent doublet, they force both doublets to bend. So this doublet, this little microprotein, this little red molecule is moving along, and as it's moving along the adjacent doublet, it's pushing them, pulling them, and forcing them to bend. So this crawling of the motor proteins bends the doublets, resulting in the whole cilia molecule bending. I have a quick little PowerPoint kind of illustrating this, and I'll put the link for this up.
Motile cilia and flagella are hair-like appendages of cells that.
I'm sorry. Didn't mean
and
to do
generate
that.
fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visual.
So here's your 8 sets of doublets surrounded by an inner set. And in here are the motor proteins.
The molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a deep.
So each one of these is a motor protein. It's attached to one doublet above and binds to one doublet below.
Detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of.
So it's going to pull it's going to pull one of these forward, causing the whole thing to bend.
Dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However,
remember the ATP is the energy currency for the cell. So you have to have ATP. This is an active process.
If dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate.
He didn't cover this yet, but there's a power stroke and a recovery stroke. So he's saying here that some of the motor proteins are stopped to allow for the recovery stroke. And we'll see that here in a moment. So when cilia are banding, when they're banding, they bend and then they have a recovery flip back motion. So they bend and then there's a recovery of a flip back motion. So the cilia bend in a rhythmic relationship of a power stroke and a recovery stroke, allowing the cilia to create a pushing motion that is repeated 10 to 20 times per second. The bending of one cilium is followed by the bending of the next cilium and the next, resulting in a traveling wave along the cell surface. Now, why do we care so much about cilia? We know that it lines the trachea. It lines the fallopian tubes. Well, we can see the major significance for it in smokers. You've heard this, everyone's heard the smoker's cough at some point. Smoking reduces the number of cilia. So there's fewer cilia to clean your lungs. As a result of the reduced number of cilia, the lungs and airways create more mucus. This creation of more mucus makes it harder for mucus to move along the track. if you can't, so your lungs, because they reduce number of cilia, as a result, to try to keep your lungs a little clearer, your lungs and airways produce more mucus. However, because there's less cilia, you can't clear the mucus as easily. So it clogs the airways and lungs and makes you cough. Also, cilia line the fallopian tubes, which helps sweep egg cells towards the uterus and promote a fertilized zygote or fertilized embryo from implanting in the fallopian tubes and implanting in the uterus instead. Because there's reduced cilia in smokers, women who smoke have an increased risk of ectopic pregnancy, which means that the embryo implants somewhere besides the uterus. Any questions about cilia and the cytoskeleton? So the primary function for microtubules inside a cell is what?
Is it A?
You're close. A is more the microfilaments.
E.
It's B. The last one, C here, is also part of microfilaments. But B is where the track, which with proteins and organelles can move within the cell. I think this might be the last organelle we're going to talk about. The nucleus is the gene-containing control center of the cell. It contains the instructions needed to build almost all of the body's proteins. It dictates the kinds and amounts of proteins needed to synthesize at one time in response to signals acting on the cell. So this is what determines what proteins will be created within the cell. Or it's the major player involved in dictating this. Cells can be uninucleate, meaning they only contain one nucleus. This is the majority of cells. Some cells, like skeletal muscle, are multinucleate because they're formed from the fusion of multiple cells together. The fusion of multiple progenitor cells together. So as a result, the skeletal muscle cells, bone destruction cells, which we'll talk about in the next chapter, and liver cells have nuclei. Anucleate is meaning the absence of a nucleus. Mature red blood cells do not contain a nucleus. They give it up and release it. As a result, anucleate cells like red blood cells cannot create mRNA to replace proteins that are gradually deteriorated. So these proteins cannot reproduce and must be replenished by progenitor cells. The nucleus has three main recognizable structures. It has a nuclear envelope, a nucleoli, and chromatin. The nuclear envelope is similar to the mitochondrial membrane and similar to the plasma membrane in that it's a double membrane system. However, the nuclear envelope is also continuous with the endoplasmic reticulum membrane of the smooth and rough endoplasmic reticulum. The inner nuclear membrane is lined with a nuclear lamina, a scaffolding of network laminins. This would be similar to the terminal web of the plasma membrane. Helps maintain the shape of the nucleus and organizes the DNA in the nucleus. Just like the terminal web, when it comes to the terminal web, remember that's made of the microfilaments that maintain the shape of the plasma membrane. The nuclear lamina maintains the shape of the nucleus. Like the plasma membrane, the nuclear envelope is selectively permeable, allowing for the passage of substances and the exclusion of others. However, the nuclear envelope is much more permeable than the plasma membrane is. The two layers of the envelope interconnect to form nuclear pores, channels that penetrate the nuclear envelope. These channels allow for things such as the mRNA molecules and the ribosomal subunits to leave the nucleus. And allows things to come into the nucleus. This movement of substances is an active transport. There's nothing passive about movement in and out of the nucleus. It's always active, meaning it always requires ATP. In the nucleus, also, there we have nucleoli. These are the sites where the ribosomal proteins and rRNA, ribosomal RNA, are combined with proteins. So this is where the ribosomes are produced. And then the ribosomal subunits and will exit the nucleus because they're involved in protein synthesis in the cytoplasm. It says most ribosomal subunits. I'm not sure why. I need to investigate a little further, because I'm not sure why we would still have ribosomal subunits in the nucleus. Because I didn't think that translation or protein synthesis occurred in the nucleus. As far as I know, it only occurs out here, which is where ribosomes are involved. I need to investigate a little further here on this. But know that the nucleoli are where the ribosomes are synthesized, the subunits. in the nucleus, also, we have chromatin and nucleosomes. Chromatin, when looked at using a transmission electron microscope, looks like beads on a thread. This bead on a thread structure is composed of 30% DNA, 60% proteins, histone proteins, and 10% RNA. The histone proteins globulate as an octamer of 8 histones. And the the DNA helix wraps around this octamer of 8 histones twice. This structure with the DNA double helix wrapped around the octamer of histones is called a nucleosome. Each nucleosome is connected with linking DNA. We are learning in research and molecular biology studies right now are looking, there's a lot of work going into studying histone function, because they play a major role in regulating gene expression. I think there's some other proteins involved, but the majority of it is the histones. Histones can be methylated or settled, or have, meaning they add a methyl group to them, or they add an acetyl group to them. When the histones have methyl groups added to them, that DNA becomes silent, meaning it's not expressed within a cell. There's no proteins created from silent DNA. When the histones are acetylated, that DNA is then open for protein synthesis. Now, why is this the case? How does this work? What is the charge on a methyl functional group? What is its chemical properties? The methyl functional group. We'll look back on chapter 2, the methyl functional group. What's its chemical properties?
The ability to form nonpolar covalent bonds.
It forms nonpolar covalent bonds. It's the only functional group we talked about that's nonpolar. It's got a neutral charge on it. What's the charge on a phosphate functional group? Somebody look up a phosphate functional group and tell me what the chemical properties of it are.
Polar.
It's polar. What's the charge on it? Is it negative or positive?
Positive.
It's actually negative. It's a negatively
lied.
charged. It's okay. It's a negatively charged functional group. Acetyl groups are negatively charged. So when histones have acetyl groups added to them. And remember, this DNA double helix has a phosphate backbone. When you put a negative acetyl group with a negative phosphate, are they going to attract or repel one another?
Say it again. I lost you. I'm sorry.
When you put a negative phosphate functional group with a negative acetyl group, are they going to?
They're going to repel.
They're going to repel. So adding a negatively charged group to histones that are also wrapped with negatively charged phosphate groups is going to cause this structure to open up. Because the DNA is repelled by the acetyl groups that are bound to the histones. The methyl group, when you add a methyl group to the histone, it's not going to repel the phosphate. And so this DNA structure is very tightly compacted. This structure with the acetyl groups is very open, or more open. If the DNA is compacted, those genes will not be expressed. If the DNA is open, it's more likely to be expressed. So the result is the extended chromatin that's been where the histone groups are acetylated is active for gene expression. Condensed chromatin, where the histones are methylated, is inactive and tightly compacted, and it stains dark. So active chromatin does not stain dark, and it's not very easily visualized. Condensed chromatin stains dark, and the genes contained within that DNA are not expressed into protein. When a cell is preparing to divide, the chromatin condenses into chromosomes. This prevents tanglement and breakage during cell division of the chromatin. If you've heard of genetic disorders where children or individuals are missing parts of chromosomes, a lot of them, it's related to a breakage during cell division. I have a little niece who has Williams syndrome, which is also called Elfin syndrome, because she's missing a major part of chromosome number 7. So DNA, before any protein is made, DNA needs to be transcribed into RNA. There's 3 types of RNA that are produced. We have transfer RNA, that are structures involved in binding amino acids for polypeptide production. Ribosomal RNA, which is part of the ribosomes that are involved in catalyzing the reaction of polypeptide synthesis. And messenger RNA, which carries the sequence by which the amino acid sequence is dictated for a protein. Now, really quickly, spend a moment Do you remember what the DNA base pairing rules are?
Isn't it like adenosine, thymine, something, something?
Okay, yep, something like that. Adenosine, thymine, guanine, and cytosine. So adenine in DNA, adenine base pairs with thymine. Thymine base pairs with adenine. Guanine base pairs with cytosine. And cytosine base pairs with guanine. So anytime you see an adenine, you know that on the opposite strand, there's a thymine. Anytime you see a thymine on the opposite strand, there's an adenine. Guanine, cytosine, cytosine, guanine. What's the difference in the bases of DNA versus RNA?
Is it the your
Uracil. And what does uracil substitute for?
thymine?
Thymine, right. So uracil is going to take the place of thymine. I have to remember what I did. Adenine. So adenine base to make an RNA strand from DNA, adenine base pairs with uracil because it substitutes for thymine in RNA. Thymine base pairs with adenine. Guanine base pairs with cytosine, and cytosine base pairs with guanine. So when in transcription, anytime the DNA is made into RNA, this is what the sequence is, dictated by the DNA sequence. This conversion of DNA to RNA is called transcription. DNA to RNA. Paisley, stop. Sorry, my dog in my lap is starting to bark for some reason. Shh, stop. Translation. Oh, actually, let's stop right there. DNA to RNA, adenine to, DNA, transcription DNA to RNA. So if I have a sequence of DNA that's AA, TT, GG, CC, TA, TA, GT, ACG. I know what the sequence of RNA is. It's UU, AA, CC, GG, AU, C, AU, G, C. Right? So this conversion from DNA to RNA, this is my process of transcription. So now I have my mRNA, my RNA sequence here. In the case of mRNA, each 3 bases here of RNA are defined as a codon. 3 DNA bases. Three RNA bases. And each codon codes for an amino acid. One amino acid, remember we have 20 amino acids. One amino acid or 3 codons code for on a stop a stop sequence, which means that's where protein synthesis ends. So a stop codon or a stop signal. And one of my codons here that codes for one amino acid codes for a start codon. It always codes for the amino acid methionine. Methionine. The conversion of this RNA sequence into protein is called translation. So translation RNA into mRNA. It's actually specifically mRNA into protein. So coming down here, DNA is encoded into mRNA, or all of the RNAs are transcribed from DNA via this process of transcription. Translation is where the information in the mRNA is used by the reading of the three base pairs, 3 bases at a time, is used to determine the amino acid sequence. Any questions about this? So DNA is transcribed into RNA. RNA, through the reading of each 3 base pairs, is converted into the amino acid sequence. Sometimes proteins outlive their usefulness and need to be degraded. Many of these proteins are broken down by lysosomes. Remember, those were the ones with all the digestive enzymes in them that function at a lower pH. Also, misfolded, damaged, or unneeded proteins in the cytosol can be marked for destruction with a ubiquitin tag, or with chains of ubiquitin tags. The name ubiquitin came from the fact that this molecule is ubiquitous, meaning it's commonplace everywhere. So proteins that are damaged or unneeded or misfolded are marked for destruction using these ubiquitin tags. When marked for destruction, proteases are attracted. Remember, ACE means an enzyme. Protease meaning an enzyme that breaks down proteins, or a proteasome which contains many proteases. They degrade small or large proteins. Any extracellular materials or any substances contributing to body mass that are outside of the cells. These include body fluids, cellular secretions, and extracellular matrix. I learned, I used to, with my second master's degree, my first master's degree, I spent a lot of time doing cell culture. And what I was trying to do for my first year was create a mouse liver cell culture. So isolating liver cells from mice and culturing them. And I learned over this very miserable, hard year that unless I plated these cells on an extracellular matrix, frequently in cell culture, you just put them on a plastic petri dish and they continue to act like normal cells, like the normal cell they were before. Well, I learned with these mouse liver cells that if I tried to put them right on a plastic, they would not act like liver cells anymore. They went through this de-differentiation where all of a sudden they were still alive, but they didn't act like liver. They quit acting like liver cells. They didn't express any of the right genes for liver cells. And I learned that the only way to get them to continue to function like liver cells after they were out of the mouse was to plate them on a matrix. Because at that point, there was a cell glue involved and a medium that allowed for those cells to signal to other cells or something to that effect and allowed them to continue their function as liver cells. If I broke up those junctions or broke up that signaling pathway, yes, the cells would stay alive, but they would quit acting like liver. So from that experience of a very long learning lesson of a full year, I learned that an extracellular matrix is crucial for some cells to function. They might not die, but they may stop functioning as they were intended. Embryonic stem cells are cells that are exposed to different chemicals, are cells that can differentiate into multiple types of cells. And they differentiate, or AKA specialize, based on different signals they receive. So these are cells that can become any type of cell based on the signals they receive. The signals can be slight differences in oxygen or carbon dioxide levels. once the cell begins to differentiate, they can release chemicals that influence the differentiation of neighboring cells. You think we talked about that acetylation and that methylation of the histones? That's key to this process. Whether a gene will be expressed or not expressed within a cell is going to determine what that cell becomes. So cell differentiation is the development of specific and distinctive structures in cells that allows them to take on specific functions. If you remember, we talked a lot about structure and function. If you change the structure of a cell takes on a certain structure, it takes on a certain function. If it develops certain structures, it'll take on the functions of those structures. So a major part of studying stem cells is embryonic stem cell culture. Isolating the stem cells and growing them up in culture and exposing them to certain signals that allow them to differentiate into different types of cells. Any questions? So on the other side of the pendulum from stem cell differentiation, we have a process of controlled cellular suicide that eliminates stressed, unneeded, aged, or excessive cells. Damaged macromolecules within a cell or an extracellular signal will activate a series of caspases. These are enzymes that are involved in cell destruction or cell death. These caspases destroy the DNA cytoskeleton and the other organelles. As a result, the cells shrink, detach from one another, round up, and start to bleb, or just start, little pieces just start falling off. These dying cells release chemicals and apoptotic bodies that attract macrophages, macro meaning big, phages meaning cells that phagocytize or eat other cells. So macrophages are attracted and these dead cells are phagocytized, eaten up and destroyed. One thing, oh, well, let's talk about this and then I'll go into that for a second. Cancer cells can avoid apoptosis. For some reason, your body doesn't recognize that that cell is strange. Most often, especially in young people, your body recognizes when a cell is going through uncontrolled cell growth. However, when you're older or in some other cases, the cancer cells avoid looking strange and don't undergo apoptosis. Heart muscle during heart attacks and nerve cells during strokes can undergo apoptosis due to a hypoxia signal. Oxygen deprivation is the same thing as hypoxia. Another thing that is important with apoptosis is we have apoptosis that's necessary and crucial for normal embryonic development. In the early development of the fetus, you have webbing in between your fingers and toes. To eliminate that webbing and give you the dexterity that you have in your fingers, there is that the webbing in between the fingers has to undergo systematic apoptosis. So apoptosis is a normal process in development. Hyperplasia is accelerated cell growth. For example, in anemia, bone marrow cells undergo hyperplasia so that red blood cells can be produced at a faster rate. Atrophy is the decrease in the size of an organ or body tissue that can result from loss of normal stimulation. In the next chapter, we'll specifically talk about the lack of exercise that leads to thinned and brittle bones. Also, in cell aging, there's a wear and tear theory that attributes to cell aging. Remember, we talked in chapter 2, we talked about free radicals being normally formed all the time. They're always formed in cellular respiration. However, normally they're neutralized. However, gradually they start to accumulate and they're not always neutralized. This results in cell aging. This accumulation of free radicals results in mistakes in DNA replication. Also, many free radicals are produced in the mitochondria because that's where cellular respiration occurs. And the result is that energy production can be diminished in these radical damaged mitochondria. Also, temporary lack of oxygen can cause blood vessels to clog with fatty materials, leading to accelerated rates of cell death. We've got 2 more slides. I want to see how quickly I can cover this. Another thing that involves cell aging is what we call the telomere clock, which is theorized to determine how many times a given cell can divide. Telomeres are found on the ends of each of the chromosomes. They protect the chromosomes from fraying or falling apart or fusing with other chromosomes. Each time DNA is replicated of a chromosome, each time the DNA of a chromosome is replicated, 50 to 100 of the nucleotides are lost from the telomeres. And when the telomers reach a certain minimum length, a stop division signal is given. So once they reach a certain length, the cell stops dividing. So the length of the telomere is the telomere clock. So cells will keep dividing. We have immortalized cell lines. We have stem cells that their telomeres don't change in shape, length. They just keep dividing. One good example of the significance of this telomere clock is what's called Hutchinson-Gilford progeria syndrome or progeria syndrome. This disease, if you're familiar with it, this is a premature aging of children caused by mutations in the gene that encodes for the nuclear matrix protein lamin A. In the proteins of fibroblasts within this nuclear matrix of cells, the telomere length in their fibroblasts is much shorter than in normal children. And the result is that the progeria hematopoietic cells don't express the lamin A with normal length telomeres, so they quit producing lamin A faster. If you quit dividing before you're supposed to, you're more likely to age. And you can't replace those cells once they're gone. Any questions? Okay, but this is the end of chapter 3. This is the last slide. You will start seeing we're going to go through chapters a lot faster from here on out. I would guess that between Thursday and next Tuesday, we will go through all of the chapter 7, and we will start chapter 12. Have a wonderful 2 days until I see you again.
You too. Thank you.
Oh, one more thing, if you're looking for the recordings, there's an error. They're not getting posted. YouTube is telling me they're too long. I've been posting them for over a year and they've always been the same length, and now it's telling me that they're too long. I don't know what the issue is. I'm going to try to resolve it. Be patient with me as I do it though. Okay.
Have a good night. Bye.
You too.
Thanks.
All right, bye-bye.
Bye-bye. Brittany and Alexa, Alex or Kevin, are you still here?
Yeah, I'm here. I just forgot to
It's okay. Okay,
click off.
it's okay.
Bye, thank you.
Welcome.